we are going to talk about the basics of plant tissue culture. Okay, let's begin. Plant tissue culture was first originated via different experiments, and this is the person who is responsible for the plant tissue culture for very beginning. His name is Haverland. Now he was the first to consider the culturing the cells aseptically in the nutrient solutions, and then saw that the plant cells are having the enormous capability of providing the whole new plant from a single cell which is isolated or collected from any region of the plant. Okay, so that is for the future times it was known as the totipotency of the plant cells. Now, plant tissue culture is a practice used to propagate plants under sterile conditions, often to produce clones of different plants. Now there lies the advantage of plant tissue culture, because we can generate the clone of different plants. That means from one plant we can have millions of the same type of plants having the same type of characteristic. If we want, we can vary the characteristics during the time course. So this is our advantage to work and play with the characteristics of plants. The production of exact copy of plant that produce particularly good flowers, fruits, and have other desirable traits. So these are the different advantages, actually the advantages of tissue culture mainly. Now we can quickly reproduce a plant. We can quickly produce a mature plant, and then the reproduction made easy for that purposes. Okay. The production of multiples of plants is very easy for this process. The regeneration of whole plants can be done from one single cell. It is known as the totipotency of plant cell, and the production of plant in sterile conditions allowed those plants to be moved, and they reduce the chances of transmitting diseases during the time course. Okay. Now also the production of plants from seeds can be done utilizing these techniques, and also we can clean the particular plant of viral and any kind of infections. Now these are some applications of plant tissue culture. Micropropagation, that means from one single cell we are propagating towards a million of cells, and that means a million of plants. This micropropagation is widely used in forestry and floriculture. Now it can also be used to conserve the rare or endangered plant species. That there lies the most important feature. And another time we can say that the tissue culture can be utilized to screen cells rather than plants. So this is another very important part. Then we can select a various important advantageous characteristics over the non-advantageous characteristics. For example, herbicide resistance or tolerance, or sometimes rodent tolerance, and all these things can be done very easily. Okay. Large-scale growth of plant cells in liquid culture can be done using this, and it can help us to produce a region of plant. For example, sometimes we must produce the root of the plant because some metabolite we want actually produced in the root of the plant. So we propagate the root of the plant in very higher amount, and then we can drag all those components from the root. In the similar way, if a component component of our interest is produced by the leaf of the plant, we must grow the plant. We produce the plant in such a way that the leaf is produced in higher amounts, and we can extract those metabolites from the leaf. So this selectivity can we can have during this tissue culture. Now, we can also cross between distantly related plant species. That's another important. We can produce some plant species which are rare or endangered. So suppose we are having a plant species which is very endangered, we can find only very few in a huge continent. We can take those plant and we can go for the tissue culture of that plant, and from one single cell, remember, from one single cell of that endangered plant we can grow million of different plants. There lies the advantage of tissue culture. Now we can also cross-pollinate distantly related. We have talked about this before. Now the production of doubled monoploid, which is also called the dihaploid plant, can also be done. We can do this dihaploid plant production from haploid cultures to achieve the homozygous lines, because homozygosity is the most important part. If we have heterozygous characteristic, that heterozygous characteristic can either give us the homozygous trait, sometimes it can give us a recessive trait too, because it is heterozygous. Sometimes it gives us dominant, sometimes it can give us recessive trait. But if we have the homozygous, it either would be dominant or would be recessive, so neither in between these two. So homozygous characteristic containing plant production is the most important above all of this tissue culture. And print this thing in your mind that in any kind of tissue culture, whatever you want to do, you must produce a homozygous line of plants. Okay, so we can actually do this production of homozygous plant by treatment with different chromosome segregating scavenging molecules like colchicine, right? So colchicine is a kind of molecule or chemical which can kill those bridges between the kinetochore and the spindle fiber, right? So it can break those spindle fiber. As a result, the cross the doubling of the chromosome number can be obtained. Okay. Now, as a tissue of transformation followed by the short-term testing and also genetic construction of transgenic plants can be done utilizing this tissue culture. So different layers of transgenic plant can be produced. Okay. And micropropagation can be utilized during the meristem culture, shoot culture, or root culture to produce large number of identical individuals. There lies the advantage of this total process. Then we can produce many plants from one single cell, from one very small starting material, which is a single cell most of the time. We can also produce some embryo of some typical type of plants which are getting endangered nowadays. So this is also called the embryo rescue. Okay, so we can do all these things utilizing the plant tissue culture. So it can help us to establish these enormous facilities. Okay, now let's move on. Now these are some instruments associated with the plant tissue culture. In the plant tissue culture lab, you must have access to all these instruments, like the dry heat treatment, that means hot air oven, for example. Flame sterilization also be there. For example, you can utilize Bunsen burner, or you can utilize normal flaming agents, right? Okay, also autoclave is important because autoclaving is the most suitable way in our world to sterile all the necessary ingredients from glasswares, mostly glasswares actually. Okay, and also culture mediums. Now filter sterilizations are important because we need to utilize vitamins and as well as antibiotics during this plant tissue culture. Now, antibiotics can only be filtered, utilized in the filter paper. Okay, so filter sterilization is important. And also, alcohol is a very friendly or useful thing for us. So, alcohol 70% ethanol is utilized for the surface sterilization, and there are sometimes chemical equipments called mercury chloride and other chemicals which can be utilized as a surface sterilizing agents, because the surface sterilization of the sample or the explant means the plant which we have taken and taken for doing this tissue culture, right? So this plant portion or plant tissue that we have taken, we must sterile it, because its surface is consisting of many kind of contaminants like bacteria, fungi and viruses and all these things. So we need to sterile this. So we can utilize these chemical components to sterile the surface of those explants. Now this is a whole procedure or an overview of tissue culture. Now as you can see in this picture, look at this is a schematic diagram that this plant is there. We can take up the portion of the leaf, we can take the portion of any kind of isolated cell from this plant, and we can put them embed them inside this culture media or plant culture media, tissue culture media. This media is specific for plant development, not the development of other organisms like bacteria and fungi and all these things. Now these cells will start divide and produce this whole plant. First, sometimes root generates after the shoot generation. Sometimes shoot generation is earlier, root generation is later. It will vary from one type of media to another type of media, but after all, when the root and shoot is totally purely formed, then you can take this plant from the culture media, then we put it in only simple water for several days. Then we take this plant and put it into the soil, and this soil in the very first level will be sterile soil, because in these conditions the plant is not habituated to any kind of bacterial infection. So if we directly plant them into the normal soil, it is full of microorganisms. It gives a random chance for killing all those plants. So that's why we need to put them into the sterile soil for very first time. Then we gradually transfer them into the natural soil environments. Okay, so this is the process of plant tissue culture in our way, and these are the photographic pictures of different stages of plant tissue culture. As you can see here, the plant is growing. This is also the large jars, and this is the plant tissue culture lab where workers are working on all the time. So this is all about the tissue culture.